So Pablo, let's begin.
Yes, I'm going to be brief because we are already 16:05. And most probably Ahmet has issues to do after this talk. So for me, it's a pleasure to have Ahmet Hoke, Dr. Ahmet Hoke here in this virtual series conference from the Argentinian Society for Neuroscience. I had the pleasure to meet him while doing my postdoc at Johns Hopkins University. We were both part of the neuromuscular laboratory. I will start saying that Ahmet has a degree of MD and PhD. The MD was got from the University of Hacettepe. I hope that I pronounced well the name, from Ankara, Turkey. Then he moved to the States and he did a PhD program in the University of Illinois under the supervision of Serri Silver, that is a well-known neuroscientist. Most people, will know his work on reactive gliosis in the central nervous system. Later, he moved to Canada. He was doing some clinical work, and also always in parallel, some work at the bench. Let me just put some up here. So it was the University of Alberta. And he has some work at that time with the Dr. Douglas Sokotne, also very famous, in the field of axon regeneration.
Yes.
So he currently is a professor of the Department of Neurology and Neuroscience. For those of you that doesn't know, neurology department ranks #1 and neuroscience #3 in the USA. So they are leading departments in the field. Also, he is the director of the neuromuscular division from the Department of Neurology at the Johns Hopkins University, and also is a director of the Neuromuscular Amazing Fellowship Program from the same department. What else? He also he published already more than 120 papers in the field of nerve repair, neuroregeneration, and neuroprotection. He's a recipient of several awards. Just I will mention some of them, but the most important, the Wolf Neuropathy Research Prize in 2018, given by the American Neurological Association, and also the Young Memorial Lecture Award in 2017 by the Korean Neurological Association. He's on the board of directors of the American Neurological Association, and also on the board from the Peripheral Nerve Society. He's the vice president also of the Toxic Neuropathy Consortium. His interests are related with the different models of neuropathy, chemotherapy-induced neuropathy, HIV-associated neuropathy. He was one of the first ones to develop an animal model and demonstrate the toxicity of HIV-1 GP120 on sensory neurons. He's also interested to study the mechanism of axonal damage and identifying therapeutic target for drug development. So today the talk is a challenge to human nerve regeneration, impact of aging and chronic denervation. And I think that his work is quite important, specifically to the talk of today. Chronic denervation is sometimes a factor that most basic researchers usually don't take in consideration when studying axon regeneration in rodent models. Just by the simple fact that in rodents, the distance of the axon to innervate their targets are really short, and usually you have axon regeneration with target brain innervation in no longer than a month. However, in humans, the distance are way longer, and in this is a factor that critically diminish the efficiency of target brain innervation due to chronic denervation and affecting the muscle that ultimately becomes atrophic. So Ahmet has, as I said, very important contributions, most of them related to the peripheral nerve regeneration. As I said, he established the first rodent model of HIV-associated sensory neuropathy. He also established pleiotrophin as a neurotrophic factor for motor neurons. He studied the modality of axon regeneration induced by Schwann cells. Now we know that Schwann cells has a phenotype according to the axon that myelinates, and this is a critical factor in order to improve axon regeneration. He also has a lot of work on neuroprotections. One of them was the pioneer work of erythropoietin as a Schwann cell-derived neuroprotectant. So Ahmet, again, it's a pleasure to have you here, and it's your turn.
Thank you, Pablo, and thank you, Patricia, and thank you to the SAN for inviting me to talk about our laboratory research. So my laboratory, as Pablo said, studies both axon degeneration as well as regeneration. And today I'm going to talk a little bit about the regeneration work that we've been doing over the last 10 years or so. I don't have any relevant disclosures. And the things that I want to introduce to the audience is talk a little bit about Wallerian degeneration. And I recognize that this may be a little too basic, but I wanted to compare and contrast the Wallerian degeneration in the central nervous system versus peripheral nervous system, and then introduce you to this concept of chronic denervation and why this is so important in terms of examining why functional recoveries in patients haven't been very good, and talk a little bit about the changes in the Schwann cells as well as in the target tissues. And I want to really highlight the fact that the rate of axonal elongation during regeneration is very slow, and that's really the limiting factor in humans. And then to touch base a little bit about the role of pathway specificity, because most of the nerve regeneration that we study in rodents, you're doing these nerve injuries in mixed nerves that has both sensory and motor axons. And yet we know that the Schwann cells that normally support one type of axon may not be as supportive of regeneration of an opposite type of an axon. So with Wallerian degeneration, with type of nerve injury, the functional recovery in patients really differs from whether it's a neuropraxia in which you have damage only to the myelinating segment and the axon itself is intact. This is what you see in people who, for example, have Saturday night palsy that wake up after being drunk and wake up with a wrist drop. Those patients tend to recover quickly because the compression causes demyelination. And then once this segment remyelinates, those patients recover nicely. But the compression can be more severe, lead to axon degeneration. In these cases, the recovery has to happen with regeneration of these axons. The regeneration of these can happen in the peripheral nervous system, especially with distal nerve injuries. But the biggest challenge is when you have a penetrating nerve injury that you basically lose all the guidance from the distal end, and you have to recreate this repair site and cross over this injury site to be able to regenerate. This is the one that in humans has the lowest chance of recovery, even sometimes with distal nerve injuries. So what are the things that really sets the peripheral nervous system apart from the central nervous system? The initial axon intrinsic processes, such as accumulation of interaxonal organelles, disintegration of the cytoskeleton, or the breakdown of the blood nerve or blood-brain barrier are very similar between CNS and PNS. But then the response of the supporting cells differs tremendously, and I think that really helps dictate the recovery. In the peripheral nervous system, you have Schwann cells that proliferate versus the oligodendrocyte in the central nervous system, which undergo apoptosis. This proliferation of Schwann cells leads to recruitment of macrophages that then helps clear the myelin debris in the pathway and allow the regeneration to take place. So the glial cells proliferate and support regeneration, whereas in the CNS, that doesn't happen. But if that's the case, why don't we have good functional recovery in humans? One of the reasons is this degeneration. In fact, this chronic denervation concept really came about after realizing that neurons do not have an innate inability to regenerate. And this was shown beautifully with Albert Aguaya's experiments in the 1980s, where he took peripheral nerve grafts and put it into transected spinal cord neurons or into the optic nerve or corpus callosum, and demonstrated that CNS neurons, when they're given a supportive environment, they can actually regenerate really nicely. So it's really this environment that dictates if an axon can regenerate or not. And in humans, we don't see this. And the reason is this chronic denervation. And there was a beautiful book published back in 1949 based on the experiences of trauma surgeons during World War II, where they have looked at close to 3,000 patients and recognized two things. One was, if they don't do any repair, obviously there's no recovery. And if they delay the repair, for example, a median nerve at the wrist, if they delayed the nerve repair 10 months, they didn't get any recovery into the distal nerve. But if they did the repair quickly, they would get good recovery into the distal nerve, suggesting that this delaying the repair of the injured nerve was very critical to supporting the axon regeneration. So is this a problem with the neuron? That is, if a neuron doesn't see its target tissue in a timely manner, does it lose its ability to regenerate? Or is it a problem in the Schwann cells in that distal nerve segment? Do they lose their ability to support regeneration? Or is it a problem in the target muscle that if you don't get your muscle re-innervated in a timely manner, you lose the ability to re-innervate that muscle? The first two questions were answered beautifully by back-to-back papers by Tessa Gordon from University of Alberta. And in fact, during my postdoc years between 97 and 99, I had a chance to work with Tessa. So what she had done was she really created a rodent model of chronic denervation or chronic exotomy. And how she did was she took advantage of the fact that sciatic nerve has two mixed terminal branches. So the common peroneal nerve innervates the tibialis anterior muscle here, whereas the posterior tibial nerve innervates primarily the gastroc and soleus muscle, plantar flexors. So she cut the tibial nerve distally and prevented these tibial axons from seeing any muscle for up to a year. A year later, she would come and cut this tibial nerve and suture it to the freshly cut common peroneal nerve. And basically, you're asking these chronically denervated tibial nerves to regenerate through a short segment of the freshly cut common peroneal nerve and innervate the tibialis anterior muscle. And they actually did a pretty good job. There was about 70% of them regenerated, which actually resulted in almost full functional recovery in that muscle. But the reverse experiment was very informative. What she did was she cut the common peroneal nerve and prevented any axonal contact with the Schwann cells in this segment. And then a year later, she would come and suture the tibial nerve. So these axons are now freshly cut, and they should regenerate like gangbusters in a rodent, and they could not regenerate and re-innervate this TA muscle. So that tells us that the denervation of the Schwann cells or the muscle is more important than lack of target innervation of the axons. In fact, in a series of other experiments with another graduate student, she demonstrated that the first 4 weeks of denervation in a rat actually doesn't have much impact on the ability of those axons to regenerate. And by 8 weeks, you start to see some decline. And by 3 months, there is significant decline. But by 24 weeks in a rodent, you basically lose ability to regenerate any axons. So those Schwann cells during that time undergo changes. And so what are those changes? The big thing that happens is that the Schwann cells that are acutely denervated, that are normally are going to be supporting of regeneration, they actually atrophy. So if you know chronically, this is from human biopsies actually, and so this is relatively freshly denervated nerve segment, and this is 2 years old a nerve segment, and basically you have fragments of Schwann cells that no longer has basal lamina. And that's very important. In addition to this, there's a decline in expression of growth-associated molecules such as P75 and ERB3 that are upregulated in denervated Schwann cells. There's a profound decline in neurotrophic factors in the distal nerve, including GDNF that is secreted by. And then more recently, Jessen and Mirsky demonstrated that a critical factor, a transcription factor called c-Jun, that is important in mediating this repair phenotype in Schwann cells is declined with chronic denervation. In addition, there is also an increase in expression of chondroitin sulfate proteoglycans. This is, Pablo will recognize this, this is a critical factor present in the glial scar in the central nervous system that plays an important role in nerve impeding nerve regeneration or CNS regeneration. So in the peripheral nerve, normally it's not expressed at high levels, but a chronically denervated nerve expresses it. So why, and this actually upregulation of CSPG happens very rapidly within a couple of days. So why doesn't it inhibit nerve regeneration in a freshly cut nerve? I'll come back to that in a minute, but I just wanted to highlight that this response of the Schwann cells after denervation is really different than Schwann cells kind of regressing back to a developmental phenotype. Normally during Schwann cell development, you have neurocrest derivatives that result in Schwann cell precursors, and then immature Schwann cells that wrap around axons. And then eventually you get either one-to-one relationship with your axon and become a myelinating Schwann cell, or a unmyelinating Schwann cell that wraps around multiple axons. And we used to think that when a Schwann cell is denervated, they regress to this phenotype. But actually what happens is they regress into or change into a different Schwann cell phenotype that transcriptionally is different than these guys. And this is what we're calling the repair Schwann cell. And the critical transcription factor that regulates this is really the c-Jun. And again, what the c-Jun orchestrates is that a bunch of downstream effects of things that are normally important in Schwann cell development and myelination, such as CROX, OCT2, HDACs, are actually blocked when the c-Jun is upregulated in the Schwann cells and they become a repair Schwann cell. So why is this important? Why the Schwann cell repair phenotype is so important in promoting nerve regeneration? So in most of our rodent models, when we cut the sciatic nerve and do a repair, what happens is the axon, all they have to do is cross this initial repair site and enter one of these Schwann cell basal lamina tubes, which we call Benza-Bungner, because proliferating Schwann cells create this almost like a railroad track where the axon can enter and are shielded from upregulation of CSPGs in the endoneurium, and they're provided with a substrate, the basal lamina, to adhere and grow because the Schwann cells are secreting a bunch of neurotrophic factors. And then they can easily regenerate and back to the muscle. But if you delay the repair, the Schwann cells atrophy and they no longer create this supportive environment. And all of a sudden, these axons now have to navigate through a nerve segment that is very similar to the glial scar in the central nervous system, in which these axons have to navigate through increase in CSVG expression. They don't have growth factor, they don't have basal lamina, and then they're not going to make it to the target muscle in a timely manner. So clearly the Schwann cells are important, but what about the muscle? And Tessa's group tried to answer this question. One of the big problems with her original model was that when you cut this common perineal, you're looking at the effect of both on the Schwann cells in the common perineal nerve, but also what's happening to the muscle. So she tried to answer that by creating these graft models, where if you take a tibial nerve and do a common perineal nerve graft that is freshly transected. So this is like the ideal supportive nerve regeneration scenario. Or you can cut the common perineal nerve, leave it denervated for a long time, and then graft it, so that you're looking at the effect of Schwann cell denervation. Or you can look at the effect of the tibial exotomy. So you cut the tibial nerve, leave it denervated for a long time, come back with a fresh common perineal nerve. Or you can try to look at the effect on exotomy. That is, you cut the tibial nerve, leave this muscle denervated, and then come back, put a fresh graft in front of it. The problem with this model, and basically she showed that this is the worst situation, that when the distal stump and the muscle was chronically denervated, that's when you saw the worst recovery. But again, the problem is that you have both the denervation of the Schwann cells here, as well as the denervation of the muscle. So recently, in my lab, together with a collaborator in plastic surgery, we tried to modify Tessa's model to look at this and basically use an upper limb model to tease out the difference between Schwann cell denervation versus muscle denervation. So our positive control is obviously acute repair, and basically you do the nerve transfer from the ulnar nerve to the median nerve here, and that basically tells us if the best possible outcome. The negative control is when you create both atrophy of the muscle by denervating this median nerve innervated muscle, as well as denervating the graft, the ulnar nerve that we're going to use as a graft. So you're basically putting a denervated nerve graft, or just doing the denervated nerve graft versus just the muscle denervation. And we use the stimulated grip strength in the upper limb, which has a better linear correlation with the actual number of axons that regenerate compared to the models or functional outcomes used in the sciatic nerves, such as SFI. And basically, what you're seeing here is that the positive control, that's the immediate repair, and Schwann cell denervation over the course of 12 weeks really did not, after 12 weeks of denervation, were not that different, suggesting that up to 12 weeks of chronic denervation of the Schwann cells did not really impede the regeneration. But the denervation of the muscle had a bigger impact and was very similar to the negative control. And when you look at the parameters or the things that may have mediated this, is that in the Schwann cell denervation group, which had good functional recovery, we had better numbers of reinnervated motor end plates, which is similar to the positive control, and the muscle denervation was similar to the negative control, which is, that's what you would expect based on the behavioral testing. And then this mirrored what we saw with the looking at the muscle atrophy and looking at the myofiber sizes. And then the interesting finding was that when you look at the retrograde labeling of neurons that reinnervated or regenerated, which is not too surprisingly that the muscle denervation group had actually a higher number of axons regenerate into that nerve segment. Let me put it back. And that's understandable because this ulnar nerve was not denervated. So it could support regeneration into that segment. Sorry. And similarly, that same nerve segment that supported regeneration of the axons into the nerve, but not all the way to the muscle, correlated with higher number of Schwann cell proliferation markers. The reverse was also true in the sense that the markers of Schwann cell senescence or aging were higher in the denervated Schwann cell nerve, even though functionally these did well. The number of axons that regenerate into that nerve was reduced and the Schwann cells were more atrophic. So basically, this study told us that up to 2 weeks, the Schwann cell denervation on functional outcome measures were really negligible. And in fact, we think that we may need, at least in the rodents, we may need a longer duration of Schwann cell denervation. But the muscle denervation, even 12 weeks is actually sufficient to impair functional recovery. And in fact, we have shown this in again, an unpublished work that we're putting together right now, looking at 8, 12, 16, and 24 weeks of denervation using that same model. And as you can see here is that when there is no denervation or 8 or 12 weeks of denervation, you get good recovery, but starting 16 weeks and 24 weeks, you had impaired recovery, suggesting that there's a critical period between 3 and 4 months in rodents that Schwann cell denervation is very important. So why is this important to human nerve regeneration? It really boils down to this problem, the slow rate of axonal elongation. And what I want to illustrate with this diagram is that if I have a patient who has a median nerve injury at the wrist, and which supplies this one muscle and the thumb called APB, if you do this injury, the patients actually have a fairly good recovery. They will, in 6 months, they will recover because the human nerve can regenerate about 1 millimeter a day. So in 6 months, you'll overcome the 6 inches of nerve regeneration. But if you injure the same median nerve up here, even though this axon never loses its ability to regenerate and can regenerate slowly along the length of this, by the time it comes to the forearm, almost 2 years has passed. And what that means is the Schwann cells no longer support regeneration. And more important, target muscle is probably atrophied beyond rescue. So there's 2 ways to fix this problem. One is instead of making the axon elongation 1 millimeter a day, why not make it go faster, much faster, 5 or 6 millimeters a day? If we could do that, then you don't have to worry about what's happening to the pathway or to the target muscle. Even with very proximal nerve injuries, you'll get good functional recovery. So is that possible? The main issue is really the rate of axonal elongation during regeneration is dictated by the slow axonal transport rate. And the reason for that is, you need to bring all your building materials in order to grow your axon. And the slow axonal transport, the SCA component is 1 to 2 millimeters. That's where most of the neurofilaments are transported. And these were demonstrated using pulse chase experiments in the 70s by Paul Hoffman, who used to be at Hopkins and retired now. And Paul and Lasek had shown that this rate of axonal elongation and the axonal transport go down with aging. So those are the two critical things. And I'll come back to that, why that's also important, the aging issue. So are there ways experimentally to manipulate the system to improve that rate of regeneration? And one thing that has been done going back to the 70s is this concept of conditioning lesion. So when you do a test lesion in a peripheral branch of a dorsal ganglia neuron, you get one rate of regeneration, usually 1 to 2 millimeters a day. If you do a conditioning lesion, injure the distal part of the same axon, wait about a week, which then induces a bunch of gene expression changes in the cell body, and then do another repeat injury test lesion, you get actually faster regeneration, 3 to 6 millimeters a day. So theoretically, it's possible to reprogram the neurons so that they can regenerate faster. And has it been possible? So one of the things that we know that goes up during regeneration in dorsal ganglia is actually cyclic AMP. And this is similar to what happens during normal development, where the embryonic DRG neuron that has the capacity to regenerate actually have elevated levels of cyclic AMP. But that goes down in the postnatal day around P7 in mice and rats. So one way to do this was artificially inject a long-acting and cell membrane permeable form of cyclic AMP, and Paul's group has done this, and basically this had no effect on regeneration in the PNS, but it did allow the central branches of the DRG axons to regenerate into the CNS, suggesting that this cyclic AMP plays a more important role in overcoming the myelin inhibition in the CNS. Another strategy has been electrical stimulation, and this is the work of Tessa Gordon and Tom Brushart, one of my collaborators here at Hopkins. And the bottom line in this is that the electrical stimulation after a nerve cut or crush does provide some benefit, but it seems to be primarily improves the pathway specificity rather than really enhancing the rate of regeneration. So one of the things that we know that injured DRG axons or neurons expressed after the conditioning lesion is a transcriptional factor called ATF3. And Clifford Wolf at Harvard actually made a mouse model of ATF3 overexpression and found that it had speed up the nerve regeneration, but he looked only 3 days after a crush injury. So we don't really know if this effect is sustained. And then he also made another overexpressing mouse, HSP27, which is downstream of ATF3, and again demonstrated that there was an effect on both sensory and motor regeneration. But again, these were done with only short-term regeneration experiments, and we don't know if these strategies will be successful with chronic repairs. So in my lab, we tried to do something slightly different. We looked at, again, we did a drug screen of about 2,000 compounds using primary spinal cord explants as well as primary DRG explants, and looked at the compounds that promoted outgrowth above and beyond what normally happens. And out of this screen, we found that one of the compounds, which was in the screen, was the C32, is actually D-cycloserine. And this compound, D-cycloserine, is actually an NMDA agonist. And we thought, let's test it in the animal model. And again, we used the upper limb median nerve repair model. And as you can see, that there is an enhancement of regeneration compared to a control. And again, we didn't do a chronic denervation model. This was really just an acute transection and repair. And we could get a little bit faster and better regeneration in terms of looking at the grip strength. And this resulted in a dose-dependent manner, higher number of axons when you look at the axon numbers, as well as electrophysiological, looking at the compound motor action potentials. In vitro, we've tested NMDA blockers to see if this effect is lost, and we don't see that, suggesting that this effect is probably independent of the action on NMDA. We're still working on trying to understand its mechanism of action before we publish this, but I thought I would show you this unpublished data. So what about other strategies to promote faster regeneration? I mean, this is one thing that I tell my patients based on this work as well as others in the field, especially Art English at Emory, who's published a lot on the role of exercise in promoting nerve regeneration. So we took mice and again used the same median nerve repair model and put them on a treadmill starting about a week before the nerve repair. And then we asked, do they recover faster compared to the non-exercising mice? And we kept them exercising throughout the whole study. And as you can see that the mice that exercised had an earlier recovery of the grip strength, as well as this inverted wire holding test. And more importantly, their electrophysiologists supported this, that they had a higher CMAP levels, and they had a higher number of axons compared to the no exercise group. Both the axon number and diameter were better, and the muscle showed less atrophy in the exercise group. And this correlated with increase in serum and tissue levels of three nurture factors that we looked: GDNF, IGF, and BDNF1. We don't know which one of these is important, and maybe it's a combination of different growth factors and perhaps some other factors that we don't know yet that promotes better regeneration. But we think exercise definitely plays a role in speeding up nerve regeneration. So I want to switch over and talk a little bit about the role of aging. So this is a paper that came out of Clifford Wolf's lab around the same time that we were also studying role of aging. And we both came to the same conclusion. So previous work from the 70s had shown that nerve regeneration in older mice or rats was slow. But we didn't know if this was, again, an effect of the aging of the Schwann cells or aging of the neurons. Clifford's group has shown very nicely in this paper that the older the mice are, they have delay in regeneration with looking at the pinprick score or the toe spread score in terms of the motor function. This was basically due to the neuron extrinsic factors. And what I mean by that is that he used a grafting model where he took a nerve graft from young to old, old to young, and looked at the pinprick score here, as well as the number of axons that regenerate into that graft segment using a Taiwan mice. And basically you're seeing is that if the graft is old, these two red bars, irregardless of if the host is a young animal, you have delayed regeneration, suggesting that, again, it's the environment that really dictates the success of regeneration. So even an old neuron doesn't lose its ability to regenerate. It's the old Schwann cells that impede regeneration. And basically in this paper, it showed that the Schwann cells in the distal nerve segment from the aged animals were dysfunctional. They did not upregulate P75 as rapidly. They did not recruit enough macrophages, and the myelin clearance was delayed. And we think that is the reason why the regeneration is delayed in these models. So we were doing the same set of experiments, but in rats. And we looked at the role of Schwann cells and the macrophages a little bit in more detail. And I want to share that with you because we looked at not just the number of axons that made it to the graft, but you also want to know how many of those actually make it to the distal segment. Because again, the distal segment is most important. You know, they have to grow through here, go into the distal segment, and then eventually make it to the target muscle. And basically what you're seeing here is that in the young rats, these two graphs, if the graft is old, you have reduced number of axons regenerate, both into the graft as well as into the distal stem. But if you have an aged rat, and you use aged graft, you again impaired regeneration to the graft or to the distal stump. But if you use a young graft and an old animal, you get decent regeneration, regenerating axons into the graft because the graft is young and has Schwann cells that support regeneration, but then they don't make it to the other end. So in this situation, you know, you can have these axons grow into the young graft, but they don't grow out the other end. And you may ask, you know, why do you have more axons compared to the contralateral side? This is very typical. During normal regeneration, the axons will have multiple sprouts. In fact, it's about 2.3 sprouts per parent axon stem. So if you're looking at these short distances into the graft or just distal, you're always going to have higher numbers compared to the contralateral side. And similar to Clifford's group, we also found that the myelin debris was poorly cleared in the aged animals. And this was true for you know, if you look at the distal graft, the age of the animal is very important. The young animals in the distal part cleared the graft, cleared the myelin debris. But if you look at within the graft, again, the ones that did the worst were the aged rats. And this, you know, impairment in Schwann cell or myelin clearance was probably due to both the Schwann cell's age as well as the lack of macrophage recruitment. As you may remember, Clifford's group had shown that the recruitment of the CD68 positive macrophages was delayed. And we saw that the same thing in the age graft, both the Schwann cells and macrophages had impaired phagocytic activity, whether you used microsphere beads or myelin fragments. And all of this correlated with inability of those aged Schwann cells to secrete necessary cytokines to attract macrophages and create this environment that supports regeneration. So a couple of those very important ones is IL-6 and CCL2 that recruit macrophages were not upregulated in aged animals. So one of the last point I want to make about the regeneration is that you know, most of the regeneration that we study is in mixed nerves. So how do the axons know to grow to the correct target? You know, Many of you may be familiar with patients who get Bell's palsy. Their axons grow, but then they may grow into the wrong places. So a motor axon can grow into the lacrimal glands. And so when patient is eating, they get tearing in their eyes and vice versa. But very old experiments have shown that the motor axons actually had an ability to find the correct pathway and innervate the muscle rather than just go regenerate or reinnervate the skin. And, Cajal almost 100 years ago had attributed to this into some sort of a neurotrophic influence, thinking that it was coming from the distal, end organ. But in humans, the end organ could be many, many centimeters away. So there's no way that it's going to be able to influence the regeneration of motor or sensory axons until they get very close to the target. And in fact, Tom Breschart, what I consider really a seminal paper, demonstrated this beautifully in the femoral nerve repair model. So your femoral nerve has two branches. One is the muscle branch that goes to the quadriceps muscle, and then the saphenous nerve that is the pure cutaneous sensory nerve. So if you do a repair, and then you can also allow these axons to regenerate all the way to the end organ to look at the impact of the end organ, but also you can cap these nerves and look at the effect of regeneration through these isolated motor or sensory branches. And what he showed was that, if you look at the early time points after that femoral nerve repair, and the way he did this experiment was he would retrograde the label from the muscle branch or the cutaneous branch. And he showed that at the beginning, the axons really don't know where they're going. Some of them grow into the muscle, some grow into the sensory branch, and some of them actually have branches in both because of that collateral sprouting. But as the time goes on, the motor axons know to pull back from the sensory branch, and more of them grow into the motor branch. And you see this, although a little bit less prominently, even when there's no end organ, suggesting that it's the pathway, again, that is dictating this. So what do we know about the pathway? For the longest time when I was a graduate student, we used to be taught that Schwann cells are either myelinating or non-myelinating, but nobody even thought about motor or sensory phenotypes. But if you think about it, the myelinating Schwann cells are almost monogamous in the sense that they never enter cell cycle unless there's an injury. So one Schwann cell that you make during development is going to stay with you until you die if it's a myelinating motor and sensory. And so it's logical to hypothesize that perhaps they also develop identities that is relevant to the axons that they're supporting. So can we study this in a more detailed manner? And so Tom and I did a series of experiments where we wanted to isolate the impact of pure motor Schwann cells versus pure sensory Schwann cells. And the reason for this experiment, which wasn't a replication of his femoral nerve model, was that if you look on this model, I said this is the muscle branch. If you think about it, even though it goes to the muscle, about 50% of the axons in a muscle branch are actually sensory axons. They are the sensory muscle spindles and so forth. So it's not pure motor Schwann cells. So to create the pure motor Schwann cells, we actually use the same model, but took ventral root grafts. So the Schwann cells from the ventral root are pure motor Schwann cells. They never saw any sensory axons. And basically what you can see is that, and we can also do this with looking at the regeneration of pure motor axons or sensory axons. growing into the ventral root graft or the sensory graft. And the bottom line is that the motor axons love to grow into the ventral root that has the sensory Schwann, motor Schwann cells, and the sensory axons grow into the cutaneous graft, which is supported by the sensory Schwann cells. And in fact, in that paper, we demonstrated that perhaps part of this response was the differences in the type of neurotrophic factors that they were secreting. In fact, perhaps not too surprisingly, given that the sensory neurons are much more heterogeneous in terms of their identity, we saw many more different type of neurotrophic factors that were secreted or upregulated in denervated cutaneous nerve, but not so much in the ventral nerve. And some of them were more specific for the ventral nerve, such as pleiotrophin and GDNF. And then we asked, is this an immutable property of the Schwann cells? In fact, what we did was we kind of forced the sensory axons to grow into a ventral root graft. And basically, we waited 1 month of reinnervation and regeneration. And you're forcing these sensory axons to grow into the ventral root and then cut it and then look at the growth factors that are upregulated. And basically, if these were pure motor graft, motor Schwann cells, they should not be upregulating any of these guys, the cutaneous predominant. And what we saw was that they did upregulate some of the growth factors, such as BDNF, HGF, but not so much of the others, suggesting that perhaps it is possible to change their phenotype. And in fact, this influence seems to be epigenetic because we can culture Schwann cells in the dish and from ventral root or dorsal root through multiple cell cycles. And for the first 2 weeks, they kind of maintain that identity that the ones cultured from dorsal root upregulate BDNF, whereas the ventral root ones upregulate the pleiotrophin. And this is maintained through 2 weeks of culturing. But you lose this after 4 weeks of culturing. And why is this important? Because in surgery, we're asking the surgeon to take a sural nerve, which is pure sensory nerve, and try to support regeneration of motor axons when they put it in a median nerve or an ulnar nerve. So in ongoing experiments, we're trying to manipulate the methylation state of these Schwann cells to see if we can actually convert sensory Schwann cells into motor Schwann cells. And this seems to work in vitro, but we haven't been able to successfully do it in vivo yet. I'm gonna skip this because this is, again, further experiments demonstrating that the Schwann cells are even further complicated in terms of their identity, depending on if they're close to the cell body versus distal. And basically, we've kind of learned that Schwann cells are extremely heterogeneous, that there are differences between Schwann cells from different subtypes of sensory axons, as well as there's heterogeneity along the length of the axon. Might just, how are we doing time-wise? I think I have only a few minutes, and I want to skip this because this is all published work, and again, using the motor versus sensory identities to look for growth factors that supported motor regeneration versus sensory regeneration. And we've shown that osteopontin was motor-specific growth factors secreted by motor Schwann cells, whereas clustrin promoted regeneration of sensory axons. So I really want to bring back to the humans and really emphasize that the distance and this rate of regeneration is really what limits success. And, when you talk to developmental biologists, often we think of, and I did my PhD in developmental neurobiology, we used to think that regeneration is really a recapitulation of normal development, but that's not the case, because during normal development, you're basically asking these axons from the spinal cord grow all the way to the target muscle or skin, and then, but most of that growth during the adulthood happens with elongation. So these neurons never grew their axons without any target for more than a couple centimeters. Because often you make it to the target and then you stretch your axon. That's how your axons become 1 meter long. But in an adult, we're basically asking these axons to kind of create a program to find their way all the way to the target and grow over very, very long distances that they never had to do. And so I think we really need to rethink this issue of either figuring out a way to support the target and pathway, or figure out a way to make the axons grow much faster. And one of the limitations is that, especially doing clinical trials, we have no way of knowing if this axon that is growing here is going to make it to the hand if I'm testing a drug, unless I wait 2 years or 3 years to see if it made it there. So what if you can figure out a way to see if you can speed the regeneration? And there's actually 2 human models of nerve regeneration that was developed by one of my colleagues, Michael Polydevkis. You can actually apply capsaicin to the skin and then come back and do repeated skin biopsies and look at the rate of reinnervation of that skin segment. Or you can do an excisional biopsy and look at reinnervation of this segment. So you can easily, within a couple of months, test the drug to see if it is speeding the regeneration. The other new development is really trying to figure out a way to improve the imaging. And in fact, this really was pioneered by Pablo's old mentor, Dr. Kazım Sheikh. So with that, I wanted to stop and thank the people in my lab who did this work. Most of these are past postdocs, my collaborators, both within Hopkins as well as at other institutions. And I want to thank the funders. Thank you.
Thank you very much, Ahmet, for this wonderful talk. I have a couple of questions, but I will ask the people to ask a question first. I don't know, we can write it in the chat. In the meantime, I don't know, Patricia, if you can read I cannot see the chat now, but if you can read the questions that appear in the chat. In the meantime, I will start with the first one. Regarding the peripheral nerve graft model, have you think about comparing the profile, gene profile of exogenous versus resident macrophages? Because in the model in which you have the graft from the young mice on an old animal, what you see is that the influence is restricted to the graft and not, the effect is not as high on the distal stem. So then there you have 2 options, or either resident macrophages or either Schwann cells. You have data for both of them. That's one question. And the other one is whether you think about doing a bone marrow transplant.
Yeah, so those are very good questions. I didn't go into the detail, but Clifford group tried to do symbiosis experiments where the parabiotic experiments where they sutured young animals to old to tease out the effect of the macrophages. And the answer wasn't very clear cut. Clearly, the most of the macrophages that in a nerve that is cut is actually blood derived. That is, they come from the bone marrow. Yes, there are resident macrophages, and yes, they proliferate, but the majority of them are coming from the outside. So if you have an aged host, you're going to be mostly dealing with aged macrophages. However, if you're using a young graft, the Schwann cells, because they're young, they're going to be able to upregulate MCP1 and bring in, even if the macrophages are old and may not be very phagocytically active, you're still going to be able to recruit them. And that's why the macrophage clearance in the young grafts, even in an old animal, was actually sufficient to get enough number of axons regenerate. Does that answer?
Yeah. And but the bone marrow transplant, have you think about it? Or?
So we haven't done that. I'm not sure if anybody has done that, trying to do bone marrow transplant between young versus old, because I think really the limiting factor is the Schwann cells and their ability to recruit the macrophages. I don't think the age of the macrophages are that critical.
Bien, okay. So I don't know, Patricia, you have some questions?
Yes, Diana has a question. Diana Jerusalinski. You can do it directly, Diana.
Yeah, so I can see the chat. So the question is, can you tell us about your ideas on the D-cycloserine mechanisms? That's a very good question. I have no idea. That's the short answer. We thought it would be the NMDA. There are some reports of NMDA receptors being present on sensory neurons as well. But when we didn't lose the axon elongation effects of D-cycloserine on DRG neurons, when we had two different NMDA receptor blockers, we thought perhaps it might be something totally unrelated. Whether it's related to metabolism, I have no idea. So the L-cycloserine doesn't do that. So it's very specific for the isomer.
Okay. In the meantime, I have to think about proteomic studies on exercise mice, looking for in plasma for molecules, I don't know, like trophic factors or something like that. Any difference that?
If I had the funding, I would love to do that.
Okay.
Because it's got to be something blood mediated.
Yeah.
And the way to prove that the blood mediated effect might be actually to do that parabiosis experiment, but you have to figure out a way to exercise only one mouse and do the injury on the other one that you're not exercising and see if it speeds the nerve regeneration.
Yeah. The other question is like regarding to blood innervation. Like I don't know, how do you think that will impact when you lose the nerve of the axon? You will lose also, depending on the lesion, the blood supply. How the blood supply from the nerve will affect the muscles itself.
Yeah, so that's a very good question. The only good thing I would tell you is that the nerve blood supply is actually almost like a plexus. There isn't a single blood vessel that runs the whole length of the axon that supply. So when you cut it, you don't lose it. In fact, when you cut the nerve and do a repair, the distal segments has almost an increase in blood supply immediately. And this was one of my first papers as a postdoc, actually, looking at the role of VEGF in promoting this in kind of a neovascularization of the distal nerve stump. So clearly there is ample blood supply in the distal nerve. That is not a critical factor. But with chronic denervation, you lose that blood supply because Schwann cells don't upregulate VEGF long term. So the VEGF upregulation, very similar to the NGF and GDNF, it happens very quickly. Within a month or two, they go down.
Okay,
We
have some questions there. Yeah.
Other questions?
Yeah.
One from Ramiro Quinta.
Yep,
Ramiro,
I
do
see
you
that.
want to ask him?
Yeah.
Or?
So the question is, can you tell me about the relationship between axon regeneration in the CNS versus the PNS regarding the myelination? So I don't know exactly what you're asking in terms of like remyelination after the injury. As I mentioned, in the peripheral nervous system, the Schwann cells proliferate and they are able to remyelinate the axons that regenerate very efficiently, as long as it's not a chronic injury. In the central nervous system, the oligodendrocytes undergo apoptosis. You lose almost 70 to 80% of your oligodendrocytes. So they need to be repopulated from the OPCs. And that's an inefficient process. And they don't remyelinate very well. And this has been studied both in the spinal cord injury as well as in the optic nerve regeneration models. Does that answer the question?
Ramiro, you can speak directly.
Sorry. Ramiro, did you say something? Okay, Mario has another question.
Hello, sorry.
Okay.
Okay.
You are there.
Thanks, Dr. Hoke. Yes, I mean, you answered my question, but I have another question regarding for example, in spinal cord injury, when the axons are cut, in some case, there is a migration of Schwann cells to central nervous system to improve the level of regeneration, for example, of corticospinal tract. So my question is related to the different kinds of myelinization by oligodendrocyte and Schwann cells in central nervous system, in particular in the spinal cord injury case. Thanks.
Yeah. No, that's a very good point. In fact, you're probably familiar with the work that the Miami group has done with Schwann cell transplants for spinal cord injury. You know, they've even done some transplants into patients. So there is ample data to show that the Schwann cells, when they're transplanted into the injured spinal cord, can actually myelinate. But they don't lose their identity. They myelinate like a Schwann cell. They still make basal lamina when you look at the cross-section of those. We have a small paper that we published a couple of years back. It was a side project, and one of the graduate students in the lab asked, can you take oligos and put it into the peripheral nerve? And what do they do? And in fact, it was interesting that oligodendrocytes do have the capacity to myelinate the peripheral nerve axons when you transplant them into the peripheral nerve.
Okay, thank you very much.
Mm-hmm.
Mario,
so,
do you have another question there from Mario Lido?
So can you further modulate the DNA methylation to improve regeneration? Yes. People have looked at the HDAC inhibitors in terms of, you know, modulating nerve regeneration. Most of that work is in terms of preventing axon degeneration. I'm trying to remember if I've come across any HDAC inhibitors and their effect on nerve regeneration directly. I'm not sure if that experiment has been done. You know, typical HDAC inhibitors and seeing if it has any impact on a peripheral nerve regeneration. Again, the big problem with the peripheral nerve regeneration is really you have to use either a large animal model or create some sort of a chronic denervation model to study, because you know, rats regenerate so efficiently that if you have a drug that improves that regeneration only a little bit, you may not see a big effect size.
So in the meantime, I will do an advertisement for Ahmet. He's the, I think I forgot
I
to
have a
mention that he's the
question
editor-in-chief
also,
of the journal Experimental Neurology. Okay. Yeah, no, I'm just saying that he's the editor-in-chief of the Experimental Neurology, and he's always open to receive top papers, in the journal.
Yes, please. So if you have good papers that's modeling human neurological diseases, especially mechanistic studies using animal models, feel free to send them my way.
I have some questions for Ahmed. Well, it was an excellent talk. Thank you very much for your presentation. I was very surprised about exercise, the effect of exercise upon regeneration. It was very effective and very quick. It was very surprising. But referring to the graft experiments, when you make a graft from all Schwann cells, these Schwann cells were also repair Schwann cells. How long was the injury made there?
Yeah, so that's a very good, so they become repair. The moment you take the graft from the old animal and put it into a young animal, the axons degenerate and you do repair. It's going to take a couple of days for the axons to cross that initial repair site and enter. And by that time, the Schwann cells have been denervated for about 3 days. And so they are repair Schwann cells.
Oh, okay, but they were not from the distal stump of an injured nerve. They were normal Schwann cells.
Yeah, freshly cut. We didn't pre-denervate them, yes.
But have you evaluated the difference between repair Schwann cells from old animals and from young animals?
Yeah,
after an injury, no fresh, no cut fresh, but also with the denervation of the nerve.
So after a period of denervation, that's a very good question.
Yes.
No, we haven't. We've looked at the different time points. I'm sorry. We looked at different time points after injury up to 2 weeks, between young versus old, and the c-Jun expression is down, lower in the old animals, during that initial phase when we expect to see a big c-Jun upregulation. And those were very similar to what Jessen and Mirsky also published. They have a very nice paper recently showing that if you can upregulate c-Jun in chronically denervated nerves or in older animals, you can actually improve regeneration and make the Schwann cells a younger repair Schwann cell. I should probably add some slides from this recent paper. It just came out a couple months ago. Beautiful paper. They created an inducible transgenic mouse where you can upregulate c-Jun with tamoxifen.
And did you see any change in semic upregulation?
We haven't looked at the MIC.
Okay. Okay, thank you. Is there any other question for Dr. Hoke?
I have my last question, yes, regarding the proteoglycans. It's just the last one, I promise. I think that it's a field that was somehow, sub-evaluated somehow, because many people work recently on the role of different proteoglycans on axon regeneration. While you were speaking, I pulled up a class that I had a presentation, and I got to refresh my memory, and it was a paper from Groves et al. From 2015 that actually was published in Experimental Neurology. You were the editor at that time. And they were looking for the role of chondroitinase ABC. But then if you see the protein index, how it's affected by different enzymatic treatment of the nerves that remove different proteoglycans, you see that most of them contribute to inhibiting axon regeneration. So what do you think, like a combination of different enzymatic treatment in order to improve or degrade all these proteoglycans?
Yeah, so that's a good point. So most of that proteoglycan upregulation is really relevant at the repair site because you really have a fibrotic glial scar there that you need to overcome when you do the nerve repair. And David Muir had, these are like 10-year-old papers, more than 10 years perhaps. He tried both chondroitinase ABC, keratinase, and one more thing, and they all had equal degrees of effects on the nerve regeneration. Basically, it helps that couple days of delay so you can get through that injury site faster, and maybe more number of axons can grow into the distal stump. But overcoming the endoneurial increase in proteoglycans and the distal nerve all the way to the target, that's going to be more difficult to overcome because you have to treat it constantly with some sort of enzymatic degradation.
Mm-hmm. But it looks like no one, would like to do the experiment in which you will test the additive effect of all this treatment, because it's not challenging. Probably not, but it's not going to be easy to publish because everyone already published individually the contribution of each proteoglycan. But from the clinical point of view, I think that's really, really relevant because there may be an additive effect.
I think it really boils down to, even if you do see an improvement, I think it's not going to affect the rate of regeneration. It may just speed the target innervation by a couple of days, but that's about it. So the effect size is going to be small, and that's probably why not that many people is working. And even in the CNS, those chondroitinase ABC experiments are falling out of favor. With the work that has shown that CSPGs are not strictly inhibitory. It all depends on the balance between the growth promoting like laminin concentrations versus the CSPG concentrations. Glial scar is not all bad.
Yeah, thanks.
Yes, but did you see the composition of both glial scar from peripheral nervous system and from central nervous system? The composition in proteoglycans is the same?
Just basically looking at some candidate molecules, similar, but there could be differences. I mean, this is the type of experiment that if one had the money, could easily do proteomics analysis of a glial scar in the spinal cord injury versus a peripheral nerve injury and compare them. I mean, as far as I know, nobody has done those experiments.
Okay. Well, I don't know if there is any other question for Dr. Hoke.
Okay. Thank you very much for the invitation. This was, you guys all enjoy your weekend.
Thank you very much, Ahmet, for your time.
Your talk.
Okay.
It was a pleasure to receive you. And next time, we hope in person.
Hopefully.
Next
Take care.
time. So,
okay. Thank you, Ahmet.
Thank you very much.
Yeah, bye-bye.
And see you. Thank you to all of you for participating in our lecture. It was the last lecture of this year, and we hope to see you next on March. Okay?
Okay.
Thank you very much.
Bye-bye.
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 Bye.
Thanks a lot.
Bye-bye. Thank you.
Nice weekend.
Bye-bye.
Thank
se
you.
fue.
¿Se fue?
Sí, sí. Está ya
Entonces, que
cansado.
tengan un feliz año para todos y nos vemos.